So what is proteomics? Well, proteomics is the study of all the proteins in the cell, or the proteome. However, this is a very complex thing to study because, well, we have over 1 million proteins in the cell. So one technique that we can employ is called bottom-up proteomics. And this involves cleaving the enzymes first with cleaving the proteins with an enzyme first to get smaller peptide fragments, which can be easier analyzed against a database. So since these proteins are so large and highly complex, well, how are we going to do this? Well, we're going to employ biology itself and use enzymes to cleave it up into smaller peptides, and this can decrease the analysis time as well as the cost. So it's a win-win for the researcher. So one tool that we've used is an immobilized enzyme microreactor, or an IMER. And basically what this is a thin capillary tube that resides in a capillary electrophoresis machine. And within this, we kind of put a molecular glue called 4-triethoxysilyl-butanoic acid. And what this does essentially grabs the proteins out of solution, immobilizes them to the inner wall, and allows us to just flush substrate over it, cleave it into the smaller peptides, and then run that against a database to see what we have in that sample. So this process is something that's fairly novel, so we had to go through many of the steps to actually get this to a point where we can pass it through the capillary and immobilize it. And we'll hear a bit more through this with more here. So I've been able to optimize this online workflow, making the immobilization a bit cleaner, a bit faster, and a bit easier for the researchers to do, which is a bit of a hole in the literature that existed before. And with that, I intend to pass it on to mass spectroscopy analysis, identify all the proteins in a sample. And one use that I'm looking to actually apply this to in the future is studying the equine proteome to look for biomarkers for equine Cushing's disease, which is a disease that affects horses with a pretty dramatic phenotype. And there's not really a good diagnostic test that's used at the moment. So with this, we're hoping to find a multi-protein panel that can better diagnose these animals, both earlier and get better treatments for these animals in the end. So if you're more interested, you can check out my poster at the end of this. Thank you.